como que llegara algo más de gente antes de empezar, pero ya son casi las 8:05, entonces vamos a dar comienzo por respeto también a los que están a tiempo y a los conferencistas. Nuevamente, a pesar de que ya lo he hecho pues, en las 2 ocasiones anteriores, les voy a contar un poquito acerca de ellos. Y en general, pues en primer lugar, quiero decir que para mí ha sido muy agradable poder tener la visita de ellos, poder conocerlos. Hemos tratado de hacer esto desde hace un rato, antes de la pandemia, desde el 20, y por fin se pudo dar. Y personas las que podríamos llamar, de hecho, como expertos en esos temas son ellos, o sea que para mí es muy, es un honor, es muy bueno tenerlos aquí. Y me alegra mucho pues, que hayamos podido tener también buena asistencia generalmente. Para que no nos ocurra lo de ayer, voy de antemano a decir que después de la charla del Dr. Weaver, de pronto vamos a dar máximo 5 o 10 minutos de preguntas. Podemos esperar hasta el final, si tenemos más tiempo, de pronto podemos hacer preguntas a ambos. Pues si no puede, él está disponible para que le pregunten pues, individualmente, para no hacer esperar demasiado al Dr. Weaver, porque nos cansamos también mucho más alargando las presentaciones y porque tenemos más o menos un tiempo definido para hacer la transmisión. Las charlas también hemos tenido la ayuda de la Corporación de Ciencias Básicas Biomédicas para transmitirlas y están siendo grabadas, o sea que es posible que más adelante puedan accederlas. Y básicamente todo. Ya a la hora de las preguntas también al final, vamos a pedir que o sea, funcionen aquí, la funcionamos en micrófono, vamos a tener dificultades para que escuchen los que están por fuera las preguntas. Y bueno, sin más preámbulos, entonces voy a hacer nuevamente una corta presentación del Dr. Weaver y posteriormente el Dr. Munder. El Dr. Weaver se enfoca principalmente en micología, evolución, epidemiología y tratamiento de enfermedades reproductivas, de la interacción de protectores con los arbovirus y en el desarrollo de vacunas. De hecho, que lo vamos a ver hoy en día, de hecho, ha trabajado con mecanismos de emergencia viral, también de patógenos y conocimientos de los vectores de estos nuevos animales como Zika y chikungunya. Y también ha estado trabajando en el descubrimiento de virus específicos de los ciclos, tales como un llamado que se descubrió en Israel, que sirvió como plataforma para diagnóstico posiblemente para el desarrollo de vacunas, así como otros vecículos. También dirigía un centro de excelencia de aborto, que era patrocinado por el CDC, que incluía alrededor de 8 instituciones académicas. Era un centro bastante grande, no sé cuántos tenía el CDC, pero muy importantes a nivel de Estados Unidos. También se ha comentado el centro de referencia para virus emergentes para arbovirus. Les he dicho que él tiene como profesión original biólogo y música, y después hizo antropología médica y Cornell, en Nueva York, hizo biología molecular y biología en la Joya de California, en la Universidad de California, y también postdoc en la Universidad de IES, alrededor del 94, alrededor de los 90, y después empezó a trabajar con UTMB, la Universidad de Tecnología en Francia, con quien tenemos currículum que nos ha permitido traernos un programa que ya lleva 7 años, está en una segunda etapa, pero renovado hace 2 años, y a través de él se ha podido enviar alrededor de 8 estudiantes de doctorado de entrenamiento de 1 año. En el segundo semestre, que nuevamente vamos a empezar a comunicar para una experiencia de doctorado en postdoc. Y en ambas etapas también hemos estado de profesores de la Universidad Europea, del CEDES, de la Javeriana, del Centro de Medicina Tropical, ahora también de la Universidad Cooperativa de Colombia. Nosotros de la tenemos entrenamientos para servir como a su vez mentores de otros estudiantes de estas áreas, que son relacionadas todas con síndrome de enfermedad diferenciada. Es un tema muy recurrente a partir de la pandemia, porque pues sabemos que tiene muchas etiologías, como ya bacteriano, parasitario. Entonces necesitamos caracterizar mejor ese problema. Doctor Líder, si ustedes buscan referencias de internet, particularmente más de 500 trabajos en los que él ha participado, y en particular para este tema de los arbovirus, alrededor de 180, en arbovirus cerca de 255 y en arbovirus 116. Entonces, nuevamente, el doctor Líder que nos presente su tema de hoy, que es nuevas aproximaciones para vacunas contra enfermedades arbovirales. Te agradezco a él y a ustedes la presencia.
Muchas gracias, Juan. Good morning, everyone. A lot of money available in the United States for countermeasure development for biodefense. And I was convinced by my chair, David Walker, at that time that we had figured out a few ways to attenuate alphaviruses, and alphaviruses are potentially biological weapons. They were highly developed during the Cold War as weapons. That I should try out some vaccine development as part of a program called the Western Regional Center of Excellence in Biodefense by the NIH. So we started this work about 15 years ago, and I'm going to this morning show you some examples of the kinds of approaches and platforms we've developed, mostly for alphaviruses. I'll mention only very briefly one project on flaviviruses. So we set out to develop ideal vaccines for emerging arboviral diseases. So first of all, they had to be simple and inexpensive to manufacture, so they would be affordable in endemic countries. Ideally, they would induce rapid and long-lasting immunity to control an explosive outbreak, but also to control endemic disease after a single dose. It's difficult to get people to come back for a second or third dose of boosters. It would be safe, either replication incompetent or very stably attenuated. And another safety goal we had was, if it's a live virus, we did not want it to have the potential to infect mosquitoes. Perhaps an immunocompromised person develops viremia, they get by mosquitoes. We don't want to initiate a transmission chain with unpredictable consequences for the evolution of the virus. And then recently, there's been a lot of emphasis on rapid development vaccines in response to pandemics, like we saw with COVID vaccine development. If there's a rapidly spreading pandemic, you can't take many years to develop a vaccine that will be able to impact the epidemic. You have to move much more quickly. So traditionally, for most vaccine development, there's been viewed as what we call a trade-off between safety and immunogenicity. And so on one end of the spectrum are very safe vaccines that are inactivated viruses, subunit proteins, virus-like particles, etc. They're replication incompetent, so they're very safe, but their immunogenicity is usually not so good. You have to give multiple doses and boosters. On the other end of the spectrum are things like attenuated vaccines, for example, the yellow fever vaccine now, after a single dose, the World Health Organization considers you protected for life. But those vaccines can have some safety problems. Yellow fever is a fairly safe vaccine, but there are occasionally adverse outcomes, even fatal outcomes with that vaccine. So we really wanted to bridge this trade-off of vaccines with others here in the middle that are replication deficient in some ways. And so I'm going to start out by telling you about our efforts to improve the safety of our live attenuated vaccines to move safety in this direction, and then I'll come back to some of these platforms, how we try to improve their immunogenicity. So the first example I'm going to just very briefly describe, and this was what I think is the best live attenuated platform that we've developed for alphaviruses. And The first one of these that we developed was the chikungunya virus. This was done about 10 years ago. We were collaborating with a company called Virigen, including Jorge Osorio, who's well known here in Colombia. And this grew out of a project earlier where we were making chimeric alphaviruses similar to the dengue Sanofi vaccine, where we used the backbone of a benign alphavirus and introduced the structural proteins of the target alphaviruses. But we found that with these chimeric viruses, they still retained a little bit of potential to infect mosquitoes. And we wanted to completely eliminate the possibility of mosquito infection. So we developed this strategy initially just to prevent mosquito infection. So alphaviruses have a genome of about 11 kilobases here. They have 2 open reading frames. The non-structural protein open reading frame is translated directly from the genomic RNA. And then they make a large number of subgenomic messages that encode the structural protein open reading frame to make more copies of those structural proteins. And there's a very strong promoter that derives expression of this subgenomic message to very high levels. Well, the strategy that we decided to try was, it had been discovered really inadvertently in work with baculovirus expression systems that certain internal ribosome entry sequences that had been tested in the baculovirus system to express multiple open reading frames from the same RNA without having cap dependence like you would normally have for alphaviruses. Some of these sequences derived from picornaviruses. They didn't function in the baculovirus system. Apparently, insect ribosomes just did not recognize some of these internal entry sites for translation. And so we exploited that finding by taking one of these picornavirus sequences and knocking out the subgenomic promoter and replacing it with the internal ribosome entry site. So now we have a genome where both the non-structural protein open reading frame and the structural protein open reading frame are made from genomic RNA. There's no subgenomic RNA at all anymore. But this particular translation cannot occur in insect cells. At least that's what we hoped initially. And we also made a second version. The other consequence of this is that we don't have subgenomic RNA. So the quantities of structural proteins expressed is much lower now than with the wild type virus. We wanted to improve the expression of the envelope proteins for a vaccine, of course. And so we made another version where we kept the subgenomic promoter. We translocated the capsid protein gene downstream behind the iris. So now insects cannot make capsid, but they can make envelope proteins, and everything makes a lot more envelope proteins, including vertebrates that are immunized. And there was one adaptive mutation that we had to use in combination with it. So we tested both of these versions. They both worked very well. The first version was a little bit safer, probably just because there's a little bit less replication in vertebrate cells retaining the subgenomic promoter. But I'm just going to summarize the results of this platform, which was licensed to Virigen and then to Takeda for chikungunya. Unfortunately, Takeda has not developed this vaccine very much, but they produced no detectable disease viremia or any obvious changes in the brain of any kind of mice, including knockouts of interferon receptors, STAT1, et cetera. This kind of mice that are used for models of things like chikungunya, or in macaques, no disease or viremia. They protect all kinds of animals against every measure of morbidity that we have for these kind of studies after a single vaccination. They produce both good T cell and humoral immunity. We know from many studies of alphaviruses that it's primarily antibodies that protect. T cells play a minor role in protection. And very importantly, our goal to knock out replication in mosquitoes was achieved. These viruses cannot infect mosquito cells in vitro or mosquitoes in vivo, even if we inoculate them with nucleic acid. So this is, I still think, a very good live attenuated platform. Unfortunately, we don't have a commercial partner right now that's really investing in this. But we've continued to work on this, and we've made vaccines for Venezuelan and Eastern equine encephalitis as well. The second platform that we worked on is the vesiculovirus platform, and I'm sure you all are familiar at least a little bit with rhabdoviruses. Shown here, they're negative strand viruses. Vesiculostomatitis virus is actually an arbovirus transmitted by sandflies. It's been used as a model system for studying negative strand viruses for many decades. And recently it's been used as a vaccine vector. And the most famous one now is the Ebola vaccine, which was developed using DSV and licensed 2 years ago for use in controlling outbreaks in Africa. We partnered with a company called Perfectus that had really highly developed this vector. I think that Perfectus has the best version of this vector available. And what they did was they took the genome, it's a minus strand genome, but shown here in the positive sense for the order of the genes. You can see they have 5 major genes here. And what Perfectus did was they took advantage of this information developed a long time ago by Gail Wirtz and colleagues, showing that the way that rhabdoviruses regulate their gene expression is through this transcriptional gradient, where the genes the closest to the 5' end of the positive strand get made at larger quantities, and as you work your way down the genome, there's less and less, where the polymerase gene has the least expression of all the proteins here. And what Perfectus did was they played around by switching the order of these genes, and they showed that they could stably attenuate the virus by putting the genes in the wrong order. So the virus isn't making the right ratio of the proteins for optimal replication, and that attenuates it. And they also made another change that also further attenuated the virus, where the glycoprotein gene here, the glycoprotein has a transmembrane domain, so it sits on the outside of the envelope of the virus, extends inward to interact with other proteins. And if they truncated this cytoplasmic domain here at different levels, this further attenuated the virus. So the combination of gene reorganization and truncation of this glycoprotein achieved very stable and predictable attenuation for VSV. And they showed this in a number of different studies where they introduced foreign genes at different positions in that order. And they could also, for example, they did a lot of work with HIV here. They could also alter the expression of the vaccine gene based on where they put it, where they inserted it, and then they could alter the attenuation based on rearranging the VSV genes themselves. So this is a very finely tuned system. And we teamed up with Perfectus with some funding from the US Army to develop alphavirus vaccines. But we also wanted to tackle another challenge with the VSV platform, and that is, at least theoretically, if you use VSV for many different kinds of vaccines, so you go in for vaccine against virus A, that's a VSV vector, and then a year later, virus B, and a year later, virus 3, you're going to develop especially T-cell immunity to all those VSV genes that are being used over and over in different vaccines. And eventually you may reach the point where that interferes with subsequent vaccination. You start by getting an Ebola vaccine, but you develop some VSV immunity, and you come back with a Venezuelan equine encephalitis vaccine a year later. That anti-VSV immunity may prevent adequate replication of the Venezuelan encephalitis vaccine, may limit immunogenicity. So we really thought that we needed a second vector in case that problem became apparent. And so we sought to find that second vector. And we ended up using the virus called Isfahan virus. This is another example. I keep telling you over and over about the World Reference Center, how valuable this collection of viruses is. So Isfahan virus came from sandflies collected by my colleague Bob Tesh in 1975 in Iran. And it had been in the Reference Center collection ever since. When we started going through the records, looking at vesiculoviruses, We were looking for one that had several properties. It could infect people but not produce serious disease. And it was different enough from VSV that we wouldn't expect cross-protective immunity, especially in T-cell epitopes. So this seemed to have all those properties. So although it produces disease in animals and is in many other ways similar to VSV, it's present in Isfahan, we know, because a lot of people have neutralizing antibodies, but it's never been associated with any disease in Iraq. So we suspected that it produced very mild or inapparent infection, which was exactly what we were looking for. And when you look at the sequences, you can see comparing Isfahan to VSV, sequence identity here. You can see that they differ by around 40 to 50%, which we felt was probably a good level where they were close enough relatives that we thought the attenuation strategy that had been developed for VSV would work for Isfahan, but they're not so closely related that we expected cross-protective T-cell. So we developed Isfahan as a second vesicular virus vaccine, and then we developed this forAlphaviruses. So we made several different vaccines. First of all, we made a cDNA clone of Isfahan virus, shown here. Same organization as VSV. Then we took Isfahan and we inserted the structural protein of the reading frame of Venezuelan and Eastern equine encephalitis. And we translocated the nucleoprotein gene downstream to attenuate the virus. And in some versions, we also truncated the gene, the glycoprotein, as I showed you before. We used the HIV vaccine developed by Perfectus as a negative control in our experiments here. And then we also compared with VSV vaccines by making a Venezuelan encephalitis version in the VSV backbone here. And just to show you some of the data here, this is an example where we combined the Eastern and Venezuelan vaccines together in a bivalent or a blended formulation. And we tested in mice to see protection against both viruses. You can see we developed relatively robust neutralizing antibody titers after a single dose. With a booster, we get much better titers, but a single dose protects very well, at least initially. And these are just data showing you the various versions of the VSV vectors here. When we did challenge with either Venezuelan or Eastern equine encephalitis, we got complete protection with all these vaccines. Animals that received the HIV vaccine or PDS alone as a placebo for vaccination were not protected. V is a very reliably lethal model in wild type CD1 mice shown here. Eastern equine encephalitis, despite being more virulent to people, the mouse model is not quite as virulent, and some animals survive even with no protection, but we did get significant protection by this combination Eastern Venezuelan vaccine against both viruses. So this platform appears to be very successful as well. It's not clear to us what the future is, but we did move it into non-human primates with U.S. Army funding and very similar results here. I'm showing you results with challenge with Venezuelan equine encephalitis. And you're going to see similar data to these today. First of all, the neutralizing antibody titers, the viremia after challenge is one of our typical endpoints. So animals vaccinated here had no detectable viremia after challenge. These are aerosol challenges because this is a biodefense vaccine, and alphaviruses as weapons, we would expect them to be introduced by aerosol. They're very stable in aerosol form. We challenged with this strain, which was from the 1995 outbreak in Venezuela, and then the old Trinidad donkey strain, and we protected in both cases against viremia. We also monitor these cynomolgus macaques telemetrically. So they're implanted with devices in their back that measure their temperature, respiratory, and cardiac function in real time. And so what you're seeing here are the temperature data, either before or after challenge in the different groups of animals. And so what you see here is a typical circadian rhythm of temperature with minimum temperature in the middle of the night, just like we experience when we're sleeping. Highest temperature late afternoon, and that's what we see before challenge in normal animals. After challenge, about a day later, we start seeing fever in the unprotected animals that receive the HIV control, but we see maintenance of that normal temperature pattern in the animals that receive the vaccine. Same here when we use the Trinidad donkey strain, complete protection against both viremia and fever are teaming endpoints in these studies. So these vaccines induce good neutralizing antibody responses after a single dose. They do even better with a booster. They also induce robust T-cell responses against the structural protein epitopes. Both Isfahan and VSV work very well. They completely protect against viremia, fever, and fatal outcomes. The non-human primate model is not fatal, but like SARS. And we've also done similar work with chikungunya virus. So this is a very good platform. It can be developed quickly for other alpha viruses, since we know how to attenuate the backbone and we know how to design the constructs where to place the alpha virus genes. So we think this will be useful for other alpha viruses as well. Now I want to move to the other end of the spectrum, our attempts to improve the immunogenicity of the safer platforms like these here. So I'm going to mainly talk about what I really find to be a fascinating story and one of the most fun things about the serendipity of doing science, and also the value of the reference center again. And that is our work with this virus called ALOT virus. So this is another very old story involving also my colleague Rob Tesh. And it began in the 1970s when an entomologist from Israel was collecting mosquitoes in the Negev desert looking for arboviruses. And he was inoculating mosquito pools both into vertebrate and mosquito cells. And it was very fortunate. Most people still don't inoculate samples into mosquito cells, but there are some viruses that that's the only way to detect the presence of a virus through traditional means. And so he found that one pool of mosquitoes was causing cytopathic effects only in mosquito cells. The cells were fusing together. And so he suspected that there was a virus present, but he was not able to identify what it was in Israel. So he sent this pool to that time Yale University, where Bob Tesh was running the reference center. Bob did a number of experiments using serology to try to determine what kind of virus this was, but he couldn't figure it out because the problem with identifying mosquito-derived viruses serologically is that there's often a lot of cross-reactivity with mosquito components in a virus culture that make it very difficult to determine serologically what family it belongs to. And so this virus basically sat in the freezer for about 30 or 40 years, defying identification, until this guy came along, a student working with me named Farouk Nassar, who had actually worked in vaccine development at Wyeth Letterly before he came back to graduate school, but was very interested in basic science. And he took an interest in this virus and trying to figure out what it was in collaboration with some scientists from Columbia University who were doing next generation sequencing at kind of the early phases when NGS became affordable. And so we sent this sample of this virus culture to them, and they came back a few weeks later and said, this looks like an alpha virus. And they had assembled most of the genome, contig, and shown very clearly that it was homologous to other alpha virus sequences. So Farouk tried to go back to this sample and to isolate the virus using various ways. It plaques on mosquito cells. But the other thing that was discovered with the sequences, there were actually two viruses present in this mosquito pool. Another was called a Nege virus, and the Nege virus was present at much higher concentration. And when we passaged the material over and over mosquito cells, the Nege virus always won the competition and overwhelmed the alpha virus that was present. So Farouk, after trying various ways to isolate it by picking plaques and things like that, he decided the best way to do this is simply to complete the genomic sequence, to have cDNA synthesized, and to assemble a clone of the virus. So Farouk did that. He produced a clone. He also showed from the sequences that it's a kind of a mainstream alpha virus right in the middle of the genus here, but relatively distantly related to other groups of alpha viruses here. And now we know that there are more what we call insect-specific alpha viruses that are close relatives of A1 in different parts of the world. But so Farouk made this cDNA clone, and he did a large amount of research on it for his dissertation. First of all, he found out it was very fundamentally restricted from infecting vertebrate cells. It couldn't enter vertebrate cells, and even if you introduced RNA transcribed from the cDNA clone, the RNA couldn't replicate. So it had several levels of very fundamental restriction here that prevents it from infecting vertebrate cells and makes it a very safe vaccine platform. And so the next thought that came to Farouk was, back to his days in industry and vaccine development, Farouk was always thinking about translation. Whether we could possibly use this virus, ALOT, somehow in vaccine development. And the initial thought was a very modest goal, and that was, it's been known that you can make an activated alpha virus vaccine for a long time, and they're relatively efficacious. They require 2 or 3 doses like boosters, but they work pretty well. They're typically inactivated with formalin. And our initial thought was, well, you know, if we can make a chimeric alpha virus, and we had done this a lot in the past by introducing the structural proteins of another virus like chikungunya, swapping them to ELOT, and if it remains post-restricted, unable to infect vertebrate cells, maybe we'll have a virus that we don't need to inactivate. And so formalin does degrade the antigens and the immunogenicity of viruses. If we didn't have to do that with this virus, it would at least induce a little bit better immune response as a pseudo-inactivated vaccine. And so it was a modest goal, and Farouk, along with another student who came along, Jesse Erasmus, they made this clone. First of all, they did cryo-electron microscopy. They showed that the structure was identical to chikungunya virus, which was great news because that means the antigens would be in a native conformation. And they also showed that it retained this restriction pattern here, at least this part of it here. The RNA from this chimeric clone could not replicate in vertebrate cells. So we felt the safety was maintained. And so we went ahead and tested it as a vaccine, and it also replicated to very high titers, but only in mosquito cells. But anyway, this work was picked up by another student, Jesse Erasmus, and this is one of my most vivid memories from 28 years as a faculty member of ETNB. Was my first meeting with Jesse after he did this mouse experiment with this ELOT chikungunya chimera here. So what Jesse used C57 black 6 mice. He inoculated them either with the ELOT chikungunya chimera at a dose of about 10 to the 8. The same dose of formalin inactivated ELOT chikungunya as a control, and I'll explain why that was important in a moment. He also used an old live attenuated chikungunya strain called 18125 as a positive control. This was a strain developed by the US Army that's very immunogenic in people, but it's not sufficiently attenuated. It causes arthralgia in some volunteers. So it was discontinued many years ago by the Army. But these animals all received a single dose, and then they were monitored for their neutralizing antibody titers out through day 28 here. And so what you see here was completely surprising to me, and I was certain it was an error that the student Jesse had made, because within 4 days of receiving the chimeric virus, these animals were showing good neutralizing antibody responses, which increased through day 28. And they were even better than the live attenuated strain shown here, including at day 28, and better than the inactivated version of the chimera here. So I was certain that Jesse had mixed up the live attenuated strain with this one, and I told him to go back to the lab and repeat this, or check your record and see if you can find out what went wrong here. but he repeated these experiments and got the same results. He also showed that the chimeric virus induced very good T cell immunity, those CD4 and CD8 here. And then he took these animals 1 month after a single dose of the vaccine, and he challenged them with chikungunya virus, and he looked for viremia, and he found complete protection of the chimeric and the inactivated chimeric at all these time points. And the live attenuated strain also protected well, but not quite as well. And these mock vaccinated animals had higher viremia. This is the other model I showed you briefly yesterday, the foot pad swelling model. You inoculate intradermally into the base of the foot, there's biphasic swelling, and the chimeric virus protected completely against both phases of the swelling, better than the live attenuated vaccine. So this was very surprising to us, but it was reproducible. And so Jesse did some additional studies to try to understand this unexpected immunogenicity. And first of all, he did electron microscopy comparing the chimeric virus to inactivated chimeric virus. And he found that very quickly after inoculation of vero cells, the chimeric virus associated with clathrin-coated pits and entered through normal endocytic pathways for alphaviruses, whereas the inactivated virus only entered through penocytosis. It clustered on the surface and did not enter very efficiently. And he also studied binding to the surface of vero cells, and he showed that the chimeric virus bound very similarly to the live attenuated virus strain, and formalin inactivation reduced binding. So we think that a major reason for this immunogenicity, and this has been known for many years, that when proteins enter a cell through the normal endocytic pathways, they're much more immunogenic. They're presented much more efficiently by MHC molecules. And the combination of very high doses, these animals get more than 10 to the 8, which we can produce very easily from mosquito cells. The combination, along with this normal entry here, is probably a big part of the reason for this unexpected immunogenicity. We also looked very closely at safety and possibility that these chimeras could somehow find a way to replicate invertebrates. When we injected them into the brains of these interferon receptor knockout mice, this is really the most susceptible environment we know of for alphavirus replication. We found that the chimeric viruses never produced any histopathologic lesions. By way of comparison, this live attenuated strain produces some paravascular cuffing that's characteristic of alphavirus viropathogenesis. The weight change in the animals continued after these inoculations. There was no mortality. And then when we did serial passages, we harvested the brains of these animals, made a suspension, inoculated new animals. We did that 5 times. The virus disappeared after the first passage. This is probably a carryover of residual virus because we started out with such a large dose into the brain and we didn't do anything other than dilute the virus at each passage. So here's to be completely safe. Just quickly, some mouse data here. So we wanted to look at longer term immunogenicity. And in this case, we're using the A129 interferon receptor knockout mice. These are a better lethal model it's easier to show efficacy and protection against disease than in the C57 black model. So animals were again received a single dose of the chimera, the inactivated chimera, the live attenuated or mock vaccination. You can see the antibody titers very robust for the chimera. In this case, the live attenuated vaccine outperforms the chimera. And we think that that's simply because these animals don't control replication of this attenuated strain very well. So unlike the C57 black animals that have an intact interferon response and limit replication of that live attenuated vaccine, it's less controlled in these animals, so they get a very high titer of antibodies from the live attenuated vaccine. But nevertheless, complete protection by all these vaccines against the fatal outcome in this fecal model here, protection against viremia, protection against foot pad swelling, and against weight loss. So seemed to be working very well out to about 9 months after a single dose. The final critical study, of course, was back to non-human primates. So we used cynomolgus macaques like we did for the other alphaviruses I showed you earlier. This was a very small study. We took 3 animals and they received a dose of 10 to the 8 infectious units. Those units are measured in mosquito cells or sham vaccinated animals. They were implanted with the telemetric devices to measure the temperature in almost real time. And then we bled them at different time points to look at the immune response. We monitored their telemetric measurements, and then we challenged them with a dose of 10 to 5 platforming units of the chikungunya strain from Lauruni, Island. We again monitored for viremia here, and the animals were sacrificed. And so these are the key results from this study. So first of all, looking at the neutralizing antibodies response, within 4 days of a single dose, all the animals showed some neutralizing antibodies. So it's a very rapid response, same as we see with live alphavirus vaccines. And then you can see out at 28 days later, we've had titers ranging from 80 to 320 to 640, which we know from many years of experience are very robust titers, easily capable of protecting animals from any alphavirus disease. Here are the temperature data, just like I showed you before challenge. The animals are all experiencing this diurnal variation in their temperature. And then about 2 days after challenge here, you'll notice that the sham or mock vaccinated animals develop a very high but short peak of fever here. And this really demonstrates the value of the telemetry. You'll notice that this occurred at the same time when the protected animals had their lowest temperature in the middle of the night. So if we had done this experiment the old-fashioned way, where we take their temperatures twice a day, in the morning, late afternoon, rectal, annual temperatures, we would have almost completely missed this fever spike here. We certainly wouldn't have had any statistical support for protection against fever, but the telemetry, where we monitor temperature almost continuously, allowed us to detect this protection from the fever peak here. And then we also had complete protection from viremia, which is very consistent in this cynomolgus macaque model. We've since done further experiments where we took these, instead of 28 days, we took them beyond 1 year, and we retained complete protection. This experiment was just finished a couple weeks ago, so I don't have the data graph for you. But we also maintained very good neutralizing antibody titers of 40 to 160 more than a year after a single vaccination. So this platform we're very excited about because it appears to be equally or sometimes more immunogenic and more efficacious than a traditional live attenuated vaccine, but with much better safety because of the deficiency in replication in derivative cells. We're doing some more experiments to understand this immunogenicity, and I'm just going to show you a couple of them. So we've also made comparable chimeras for Venezuelan and Eastern equine encephalitis and tested them I think I got a slide mixed up here. Yeah, I don't have the one here that I expected. So I'll show you these data. This is also chikungunya. I thought I had. We've made these vaccines for Venezuelan and Eastern equine encephalitis, and the results would be very similar if I showed you those. But we also did an experiment with chikungunya. And in all 3 of these vaccines, we wanted to know how quickly we could protect animals. So I showed you that antibodies appear within 4 days. But what about even earlier? And one of the reasons we're very interested in this is that we do a lot of research at high containment at BTMB. And we have excellent facilities, personal protective equipment to protect our investigators from aerosol exposure. But we can never completely reduce the risk of a needle stick. And we regularly do have exposures through needle sticks, an average of 1 every 2 years maybe. including to things like Ebola, but also to alphaviruses occasionally. And we always want to have something on hand where we could prophylactically, but post-exposure also protect the laboratory worker after a needle stick. So we do these kind of experiments where we push the limit and we challenge animals very soon after vaccination. For example, at day 2 or day 4 after a single dose. And what I'm showing you here is the survival rate in the A129 mice. This is the lethal model again, where we show complete protection from death within 2 days of vaccination and a significant reduction in viremia here. So we don't think this is the result of adaptive immunity. It's too fast for adaptive immunity. We think that this is another clue to the immunogenicity of this vaccine, is that it's inducing a very robust interferon response after inoculation of these large doses of entry into cells and expression of presentation of pattern receptors that comes on double-stranded RNA. Perhaps that's part of the early immunogenicity of these studies. So just to summarize this work, all of these chimers, we may have 5 of them, including a Mayaro version, that also appears to be working very well. They replicate to very high titers in mosquito cells, so it's very feasible to make these doses that I'm showing you very cheaply and effectively. They're completely replication effective for vertebrates. They induce rapid adaptive immunity, always by day 7 in any animal model we've used, strong T cell responses. We can protect within 2 days of a vaccination against chikungunya Eastern and Venezuelan. And then in the macaques, we can protect through day 31 against all of these endpoints. And we just learned a couple of weeks ago that now we can take this up to beyond 1 year of complete protection in the non-human primate as well as the mouse model against all of these endpoints. We've also found another good use for this chikungunya ELOC chimera. So we work with a company called Bios. I imagine you can buy this kit here in Colombia for chikungunya serodiagnostics. And Bios work with us to use this chimeric virus as their antigen in all of their chikungunya license. So if you buy this kit, which is also one of the best performing of the kits as determined by the CDC, not just in our opinion, this will include that virus that I just showed you that we developed as a vaccine. And finally, you can also use this approach for flaviviruses. We started this work making the Zika vaccine with insect-specific flavivirus discovered in Trinidad called a repovirus. You can take the repo backbone. You can introduce the structural pre-M and E proteins of Zika virus and use this as a vaccine with very similar results as I showed with flavies. And then an Australian group led by Roy Hall has actually used several other insect-specific flaviviruses to develop even more chimeric flavivirus vaccines that also appear to be very promising. So just to end up, to mention some of the key players here, the work that I showed you really started with the live attenuated vaccines that we developed in combination with Takeda, which bought the company that we started with called Envirogen. Jorge Osorio, Dan Stinchcombe founded that company. The Chilean Primate Center did some of our early non-human primate studies before we started doing those in Galveston. Ilya Frolov and others from Alabama helped us design some of these vaccines. And Ann Powers at CDC worked with us. The structural biology I showed you for the chimeric virus was done at Baylor College of Medicine. And these are our main collaborators at Perfectus Biosciences. So thanks for your attention. And then move on if you want to take a couple questions or wait until later.
I have a couple of queries. One is, so some GA, sorry.
They put it into thousands of volunteers at very high doses, which really established the safety of the vector and allowed it to be developed faster for things like Ebola. So they simply had a bunch of these HIV vaccines that we simply used them as a negative control. So the animals received the same VSV backbone, but they didn't receive any alpha virus genes. And we wanted to make sure that it wasn't the VSV immunity that was protecting the animals. So that was simply a negative control that we used in all of our experiments.
And the result that you had for cellular immunity for a lab.
Yeah.
I saw that you had like interferon gamma measure in SANI4 cell.
Yeah, that's just part of the assay for the T cell response. It doesn't have anything to do with what happens in the animal that's vaccinated count. It's just part of the T cell response where you measure the interaction between T cells and the antigens by their response in producing interferon by those cells. Yes.
It's always for questions. First one is about these ELA virus. What I had understood was that it was replication deficient in vertebrates due to mainly its inability to enter the cells and its ability to reproduce its RNA genome within these vertebrate cells. So what I understood was that you demonstrated that it was able indeed to enter these vertebrate cells, but not maybe replicates within the cells. Do you think, or maybe did you identify a receptor which could be responsible for this, for the mechanism of the virus entering these cells, and so that maybe it could enter as well in the cells?
Yeah, so I think that wasn't really clear. So the normal ALOT virus, not the primary, cannot enter. We tested lots of different versions of it, cannot enter at all. So we think that restriction is probably the receptor interaction that's missing. Now the receptor for chikungunya and Venezuelan encephalitis has been identified. We haven't done any detailed studies like looking at binding ALOT amyloid proteins to that receptor to make sure it's a receptor phenomenon. We just want to make sure that it's safe, and so we haven't gone to that level of detail. But when you make the chimeric virus, now all those structural proteins on the outside, including the amyloid proteins, those are chikungunya proteins. And now the virus can bind to the normal receptor, we assume, and it can enter through the safe pathway. And probably those proteins that it's carrying with it are presented T cells much more efficiently than ALOT inside the cell. So what does remain restricted is RNA replication. So either with ALOT itself or with the ALOT chikungunya primer or any of the others, we take the clone, we transcribe the RNA, and we electroporate it into a variety of different cells. When we electroporate it into mosquito cells, it replicates very well, produces high titers of virus. When we electroporate it into any vertebrate cell, we see no evidence of any RNA replication, no expression of any ALOT or chikungunya proteins, no evidence of primary virus. So the chimera has, we no longer have to have safety of lack of entry, but we still have very fundamental safety of lack of RNA.
One last question that I have was, sorry.
I would like to know, taking in mind the fluorescent survival of the ISO, we know that other viruses are really restricting the amount of genes that you can put in. You know how many genes for size of the that it can come in without affecting the immune?
So with VSD, I think the upper limit is about 4 to 5 kilobases. So the open reading frames that we use are about 3 kilobases for the envelope proteins that are alphaviruses. So we're not pushing the limit. Now, for other kinds of vectors, they can carry a much larger cargo. This particular platform. So the other thing I want to make sure is clear: VSV is going to be used as a vector for many different virus vaccine targets. The ALOT vector, we have not yet come up with a way to use it for anything except viruses, because we're relying on putting in genes that allow the virus to replicate with the mosquito cells. We've tried a little bit to make fusion proteins, fuse another peptides or small proteins onto the alphavirus envelopes. And it works to some extent, like we put a flavivirus domain 3 of the envelope protein, but it reduces the replication a lot. So now we lose the advantage of producing 10 to the 9 or 10 to the 10 particles in mosquito cells. It drops to maybe 6 or 7. So we've lost the manufacturing capability. So right now it's only useful for alphaviruses.
Biología en la Universidad Federal de Río y también hizo PhD allí y sirvió pues de profesor de Reform. Él ha trabajado en múltiples temas relacionados con leptospirosis, de los patógenos y desarrollo de vacunas y modelos animales como el hámster. Y allí lo olvidé decir que también ha trabajado como editor en la revista PLOS Neglected Profit and Disease, está presente y también es miembro del IARCU, el comité pues, para control apropiado de manejo de animales y experimentación. También pueden encontrar más de 68 trabajos relacionados con el tema, si buscan en internet a su nombre, pues, los que han colaborado. Para hoy tenemos entonces la charla de él, que es de Translational Research on leptospirosis, donde field to the bench and back. Estudio que nuevamente al final de pronto tenemos que ir solamente con las preguntas, porque a la tienda nuevamente como para analizar la vida de todo más. Entonces, si existen otras preguntas, pueden consultarle a ellos después de que termine.
Buenos días a todos. Buenos días. So we're going to be back to after like I don't know why. So you know we talked a lot about left yesterday. I'm not going to go back into talking about it. I'm just going to remind you a little bit about the determinants, bottleneck, connection, and strategies that we use for preparation. It was not scheduled, but I'm going to talk also about preparation because that's my lab has given me a lot of work in that area as well. But I'm going to talk a little bit about the translational work that we've done in terms of diagnostics. So, this is what I finished yesterday, just to remind about the fact that lepto is environmentally transmitted disease, reservoir that containment prevent transmission, stool of infections, and any pathogen determinants for influenza transmission, effects of the climate change, globalization, and also the fact of the neglected disease we talked yesterday, and all the one of the foods that we keep. So as a zoonotic and disease, you can see that strain-specific factors for the pathogen actually are related to transmission. Exposures to inoculum dose or infection, about yesterday. And then, of course, we have the host date and acquired factors that are the term how your symptoms and how your outcome of the disease will be. So a couple of years ago, this paper actually showed the bottleneck is below for several diseases, and actually they used alpha-lacto interesting as a zoonotic disease, one of the main diseases that they work. And you can think about, between like all the bottlenecks that you can see for lacto, they have some question marks regarding like especially leptospirosis. We'll talk a little bit about yesterday that we were working to try to solve some of those issues. And then, between the exposure and the host barriers, we also talked a little bit about yesterday also now. But you can see the bottleneck start large for lacto, will start like accumulating when it comes to humans over. But you can think about So you can think about when you think reservoir distribution, you can work in source reduction in the environment. And then again, we talk about it. That's what a lot of the people do in terms of like rodent control, which is not very effective. And then for animals, we're going to talk a little bit more about yesterday, but then today. But you can do immunization here. You have like vaccines that are available for animals. And then again, you go back here and try to work with source reduction as well in that region, which is not easy. And then their approach to risk mitigation, that's what you have nowadays to do to avoid human exposure. And then, of course, this the best thing would be to do early detection. We're going to talk a little bit about that as well. I mean, ideally, if you have, you can see, if you have a good immunization here, you can have like a good immunization here, you can actually reduce a lot of that over as well. We're going to talk a little bit about that. So, in terms of strategies, just as a summary of what I thought, in terms of source control, you can do chemical control of rodents, as I mentioned, not very effective. Environmental control, food source and habitats. Again, you can do that, but in some areas, rural areas, not exactly very easy to achieve. Identify reservoir source, which is important, but needs studies to do that. And again, there's such a high variability throughout the world that there's not a lot of studies available. And of course, immunization is the best tool that we have. And to prevent exposure to humans, you can do disinfection of the areas. We talk about like scratches that you have. But again, I saw you the situation that happened in those areas. Not exactly an easy thing to do. As long as you do education for people to do like protections, foods and clothes and things, that's not a reality that happens in those areas. Of course, you can do various approaches in risk avoidance and in block transmission sources. And then to prevent disease, again, the best would be early case detection. Diagnostic, we talked a little bit about yesterday, it's not very good. And then you can try do, once you have detection and diagnostic, you can do treatment, which is very efficacious once you have in the early phase of the disease. And in some cases, especially like military or people in risk, they've been using chemoprophylaxis with doxycycline for people as well. So, I'm just going to tell a couple of stories that will fit in the theme here. So this is like the leptospirosis IG-like proteins. Those leptospirosis, those are together, our group together with like UCLA a couple of years ago, like 20 years ago. We discovered those proteins, and then they were like this new member of the IG superfamily of bacterial virulence inspectors. They actually found like lepto have like 3 of them, and they call like deviated BPC. They're huge proteins, several domains, with like a huge C terminal as well. They are surface exposed, they are regulated in conditions that mimic the host environment. They bind ivermectin in vitro. And they at the time highly immunogenic for patients who have been exposed to lacto. And there's at the time there's like a huge potential for both like diagnostic and vaccines. The interesting thing about those proteins, so for example, in most of the pathogens they have mediated BPC actually is like a pseudo gene, doesn't work, but individually when you knock them out, they actually have no effect whatsoever in the virulence. There's some sort of like, there's a lot of redundancy in leptospirosis in the genome. And so we know that actually B was I mentioned yesterday was the first that we used targeting against lacto, but there is no effect in virulence. More recently, a study showed that together, when you were able to delete them together, which is actually very easy and active, but they use like a fatal system and they actually deliver the motor of the protein. They do seem to reduce virulence in bacteria, but it has to be both at the same time. So as I mentioned, they became like very interesting candidates for both vaccine diagnostic and therapy. I'm going to talk a little bit about that. So in terms of diagnostic, that's the best, sort of like translational work that we've done in the field so far. So we use the LIP-B, one portion of the LIP-B that we found out that was highly immunogenic, and we developed this assay has been approved in Brazil. So it's like a joint EU-US patent, and it's like a rapid diagnostic point of care. So together with ChemBio, we do like this dual platform, so the lateral flow format. You just put the blood, put the finger, put the blood. 15 minutes later, you have your result. We did like a multi-center study, has like a sensitivity over 90%. You can see here that acute phase, 85% of people are positive. If you do less than 7 days, 77%, which is still very, very good. And of course, the convalescent phase, we have like almost 100%, and very good in terms of like specificity as well. So has been approved in Brazil, so it's actually used in Brazil. And at one point, we actually had like a SBIR phase 2, and I went to try to generate like a more applicable world. I mean, unfortunately, this actually it didn't go through, and we were talking last night about it, and I talked yesterday. This is a neglected disease. The state company, they do like a test very similar to syphilis, which is actually sold to Brazil, and they make millions of it because syphilis is important. It's a free tax. Brazil has a very good like health system, so every pregnant woman can actually do the test. They make a lot of money with that. This test has been approved in Brazil, has never been used by the public system in Brazil. So the company completely lost interest. They're not making money. So they're not very interested in the test anymore. The issue with the test is that, as I mentioned before, we only have one serovirus circulating in Salvador, Brazil. So the test is very good. The results are from Brazil. But if you try to apply that island along the other areas, you have like more diversity. The lakes are not very, the amino acid identity is not very good. So the results are not so good. So in order to make a test that's going to be a little bit of the world, you have to put like a lot of work in trying to increase the viability of the agent that you're using. And again, there's no interest. Any other company was interested in working on that. So this didn't move past this test that has been approved in Brazil, and again hasn't been used at all. I think right before the pandemic, the person who was one of the directors of the Brazilian Ministry of Health, he was, he actually was involved in those studies that developed it. So he was pushing the Ministry of Health in Brazil to actually use this test, and then eventually COVID came, and that fell, and the last president came, so the person actually left the Ministry of Health because of political reasons, and so now I don't even know if this test is going to be ever used in Brazil. And then just quickly about other diagnosis approach that we use for lepto. So the single real-time PCR is like something that I really enjoy and like to use. I mean, this actually was also developed by CDC. Actually, the lepto-32 base. Lepto-32 is a protein that's only the pathogenic species of lepto. So they tried to use in the past 16 acids. It was a lot of like a false positive in humans. So this actually has like 99% specificity, and the sensitivity varying with time. So you can see here that the early phases of the disease, we have like very high sensitivity. So the antibodies are very low. As soon as the antibodies start increasing, then the lepto actually disappears from the blood system, and then you start getting increased sensitivity of the test. So the qPCR is very good if you can get people right in the beginning of symptoms to get positivity. So actually this is very good for, I mentioned to you about all the species that we have. The pathogens are divided between P1 and P2. P1 is usually high, more pathogenic, P2 less pathogenic. So this lipid actually is very good for all the P1s. We recently, our group just like optimized, because as I mentioned before, there's so many new pathogenic species that have been discovered in the past couple of years. So we actually optimize those parameters. Actually, today it gets like every single, there's like 28 pathogenic P1, and you know it can detect every single of those 28 P1 in a single PCR. And then more recently, we've been developing multiplex PCR, because there's always this question about what is important between P1 and P2. P2 in the past was called like intermediate, which is only people inside of the fire actually assume what intermediate mean, but basically it just means that we don't have a lot of evidence about how pathogenic those strains are, although they are genetically more close pathogenic ones compared to saprophytes. So we just recently developed a test that we use the same primers in the 23S of lactose. So the foreign reverse primer actually binds both P1 and P2, doesn't bind to saprophytic species. And then we have two different probes in that assay. One is specific for P1, one is specific for P2. And so at the same time, we can actually identify not only pathogenic leptospirosis, but also differentiate between P1 and P2. And you can see, it's highly it's very good in terms of like specificity, so you can always detect P1 with P1 probe, and always detect P2 probe, and they'll detect saprophytes. And also we've been currently just evaluating clinically this, and actually that's the assay that Dr. Jennifer has used for her studies with the monkeys, and work very well, and we just evaluated that clinically to be used more sustainable in our studies. So, continue the story is, I'm not telling a story that happened when I was in the PhD a couple of years ago that basically changed my life and my career, and you're going to see why. So, I showed you yesterday a study that we've done about all the isolates, different outcomes that we've had in Brazil. So, as a part of that study, what I was doing, I was basically just cloning strains of lactose. So I get all those isolates, I'll clone them, so I can do like sequencing from those clones. So when I was doing one specific clone, it was like actually a patient that had pulmonary immune syndrome. He actually passed away. But I noticed in the play there was like a small, colonies and small colonies and large colonies. So this is the normal, colony that you see in leptospira. So and then, again, this is when I look at the microscope, you can see very morphologically the hooks there. And this strain was a little bit more straight. You can see any hooks. And then, if you look in terms of like fertility, you can see those are what we call like translational fertility. They're able to move. Those ones, they have some sort of like grounding motion. You can see they're trying to move, but they cannot. And so I was just trying, okay, so let's see what happens with this. So, just give an idea about fertility of leptospirosis in general, because it's completely different from other bacteria. So, the motor and the of lacto in the basal body is very similar to any other bacteria, E. coli, Pasteur, Salmonella, but it has like a very different structure in terms of the body of the flagella, and because it has a sheath. And also important to say is that it's very plasmatic, so it's inside the cell, not outside the cell. But that actually is very important for spirochetes, because it helps them to have high velocity in high viscosity media. So if you put lacto in liquid, it just moves. But if you put like in blood, it actually moves even faster. So it helps them to go very quickly and move in blood. So lacto actually, even within the spirochetes is even more interesting. As I mentioned, this is the shape of lacto. This actually, lacto has only like two flagellas, one in each pole, and the flagella actually, if you purify the flagella of lacto, actually coils. And this is borrelia, you can see that the flagella is actually straight. And that coil actually exerts, a huge strength in the body cell and actually makes it bend. And that's why it uses the hooks of the bacteria. And that shape of the bacteria actually makes it move. So if you have a hook the cell doesn't move, a spiral doesn't move. When you have a spiral and a hook, it just moves in that direction, and they can, that's what you see here, a spiral and a hook, and that changes in milliseconds, and they can go back and forth. So what I noticed is that the strains that I showed you, the large colonies, which is the rotile clone and the motility deficient clone, you know, when you purify the flagella bills are coiled as normal, and this one was more straight. So, you know, we decided to explore a little bit more about that. So that was interesting because this happened like, you know, almost like 20 years ago, like 16 years ago. So when I tried to push this to understand, and I talked to a lot of people working in utility, lactose, heartbeats in general, and everybody said like it's impossible for you to identify. This is like a spontaneous mutation, right? This is like a genome that has like 45 protein, 45 protein. It's like it's impossible. It could be motility, could be like infectious, could be a bunch of things, could be like several mutations. And that was a time when, like, you know, sequence of like one bacteria cost like $10,000, $20,000. It's like, you know, you're not gonna be able to find that out. Just, you know, today you can sequence a, you know, a genome of bacteria for $100. But that time was very difficult. So actually, I spent a time in Institute Pasteur. You know, I'm very stubborn. That's part of my personality. So I decided to dig into that, and actually, I was able to do like mass spec. And, you know, and to be honest, I was very lucky because when I did like mass spec, you know, you can see there's a huge band that was missing there. So we're able to look into that band. I identified one specific protein that was in that band, and then I did like a lot of like mutations. So that actually the spontaneous mutation, there was a change in amino acid, and actually that led to a truncated protein which was not expressed. We did some knockout, we did target mutagenesis, mutation, and then we in fact figured out this novel protein. It's like a we call protein, FCPA. So it has no homology with any other protein in lacto, and also not in other bacteria, not even like spirochetes. It's very unique to lacto. It's 13th most abundant protein, making lactoprotein. It makes sense. So protein is covering, and you know, the flagella. And you know, in the past, that's what people thought that the flagella of lacto were. You have the flag B's, which is like the core of the filament, and then you have flag A's, which would be the sheath. But our study actually showed that you know, FCPA is the one who is in the sheath. You can see there like the B with the antibodies in the FCPA flagella. So it's all outside. So you know, it changed the paradigm of like lactose, showing that FCPA actually was a protein that was surface in the sheath. So basically, you know, what happens here is that you have the wild type, which, you know, you have morphology, large colonies, foil flagella, a diameter of the flagella of like 20.5 nanometers in high motility. And when you have the deletion of FCPA, you know, this is the phenotype that you have. The cells are straighter, smaller colonies, straighter flagella, and then you have like a shorter diameter of the filament, and the lack of this sheath, you know, affects the interaction with the body cell, and then it gives you this phenotype. These cells are straighter. They almost like, you know, they cannot divide properly. They stay like chains, and you can see that they're moving their ends with their flagellas, but they cannot actually have the strain and the thrust to actually do the activity. So, you know, that study itself, you know, changed the world of motility in spirochetes. We actually found out later, not only has FCPA, but there's actually another one that we call FCPE. And, you know, there's a recent paper published in Life that shows that actually flagella, although it's a tubal structure, the flagellum lacto is completely asymmetric. So you have like a protein in one side and completely different proteins in the other side. So it's a very asymmetric structure. And that leads to like a novel discovery in terms of motility. I'm not going to talk a lot about that, but it's like it's a beautiful, amazing way that leptospirosis actually able to move and to infect the host. But what I'm going to talk to you about is that, you know, the interesting thing about the FCPA is that not only it was nomotype, but actually was completely attenuated. Right? So high doses of that bacteria do not cause disease in those animals. Just to see here, this is like a dose of 10 to the 8. You know, all the animals survive. If you complement back, they all die. You know, wild type die. But the wild type can actually kill hamsters with like less than 10 leptospirosis. So this is how attenuated it is. But one interesting thing that we found out is that although it's attenuated, it's still able to disseminate, and then it is completely cleared by the immune system. So especially like the liver and blood and the kidney, you can see some levels of of this protein, the FCPA, but then it clears out and you don't see anything. So we ask ourselves, okay, so why do we use that as a attenuated vaccine, right? So you have all the proteins that are still necessary for pathogenesis. You know, the only problem is like the motility. So maybe the immune system recognizes those proteins are going to protect against infection and also maybe like a more broad protection. So just to give you a story about leptospira and vaccines. So, you know, as I mentioned for you before, leptospira was first isolated in 1916 by the Japanese group of patient. And at the same time, this group actually was pretty amazing. They basically, most of the things that we know about lacto, they actually work in the next 2 years. And one of the things that they showed out at that time is that if you get leptospira, attenuated them, put in a animal model, they are protected against the same bacteria. So basically showing that there's a protection when you use a whole cell bacteria. And that actually has been the system that has been used for the past 50 years in terms of vaccines. So as I mentioned before, there's several commercial vaccines available for veterinary use throughout the world, but the system is exactly the same. So you get a bacteria, you inactivate it, and you use that as a vaccine. So the issue with that is that the antibodies that are produced is all LPS. So the LPS is very conserved among those serovirus. Remember, I mentioned to you there's like over 300 serovirus. So if you want to have a vaccine that protects against everything, you would have to get like 300 serovirus in those vaccines. Which makes no sense and not going to be feasible. But the problem is that most of those vaccines, and if you go back and look at vaccines that are available in Colombia for dogs, or cattle, or pigs, usually they have like 5, sometimes they have 7 serovirus. They try to put as many as possible based on what they think is the serovirus that are circulating or important for that species. The problem is that a lot of those studies are made in Europe and the United States. And you can imagine that whatever happened in the United States and Europe is definitely not what's happening in Brazil, and India, Colombia, Peru, or Sri Lanka. So that's the problem with those vaccines. They're not very effective. And one vaccine cannot represent the diversity of immunological settings worldwide, and there's not a universally effective in preventing care. Although there's a lot of money involved in veterinary science when it comes to leptospirosis vaccines, I mean, there's no interest of those companies to produce vaccines specific for each of those regions. And as I mentioned before, there's a lot of places that actually there's no they have no idea about the physiology of leptospirosis, so it makes it even more complicated. So the vaccine that you're using is the same that people are using in other parts of the world, and they're not very effective. So there are some vaccines for humans. I mentioned before but there is some, but they are very low. So Cuba, China, France, Japan, I think now has one. But it's only applied in that country only for people at risk, but it's exactly the same technology. Cuba actually has it's the oldest vaccine that they have, and they have several issues because they did a very good study. There's like 3 serovirus that they thought was important. They produced the vaccine. A couple of years later, they had like huge outbreaks of that, people dying. They found out that because they were vaccinated against those 3 serovirus, one serovirus that was not important before became more important, and they said, change the whole vaccine for that reason. So so those are the other things that happen too. You're protecting for serovirus, and some other serovirus that might not be important or not so prevalent, comes up and becomes an issue. So of course the goal has always been to pseudo-unit vaccine candidates, and this is a table that is actually a couple of years old, but things haven't changed. Most of the time, people use the protein that they know and they think that's important for pathogenesis, that might be important for vaccination. And what I want to highlight, because that's why I showed before, you can see that the best vaccine, the best results have been the lips. So it is still the best candidate that we have. But there's a lot of issues with the lips, as I mentioned before. Immunogenicity, protection, sensitivity to purification, solubility, which happens with all protein, but it's like a huge protein to be expressed. But the major problem is that the ligand sequence identity is only like 60 to 75% among all of leptospira species. And actually, Lig-A, which is it is the best one, and you can see almost like all the studies are 100% protection. The Lig-A is not only present in all the pathogenics, it's only like of the 28 ones that we have, I think only like 10 of those actually have Lig-A. But also the the identity is very low. So all these studies, although they have like high levels of protection, it's only homologous protection. So you have like ligation on one COR, and you can only protect against that COR. There's no cross protection to any other CORs available. And the other issue too is actually ligate, although protects 100% against lethality, it doesn't protect against colonization. All the animals are allowed to survive. They get colonized, so there's no sterilized immunity, which is also an issue when you think about animals. So, there's a huge gap in terms of prevention. So there's no effective prevention controlling the source of the setting. That's what I told you yesterday. Rural leptospirosis may even be difficult to control. There's a large number of serovars that have been a major challenge to develop a vaccine. The current ones are only serovar specific immunity. And even if you have multivalent vaccines, which are the most common ones, they are unable to achieve sufficient coverage for all the serovars that are available out there. There's a lack of correlates of natural acquired immunity both in animals and humans. And of course, the progress is widely believed to be dependent, finding effective candidates. Most of the candidates are surgically associated, host express proteins, but all the conventional approaches that have been used so far have been failed to use to try to work with this widely applicable vaccine for lapse, which is, what we call the holy grail of the field. So, going back to the hypothesis that I have for the FCPA, so we decided to use FCPA then as a attenuated vaccine. So, we only do like one subcutaneous vaccination, one dose, 10^7. We use the vaccine, which is basically the bacteria as a control, BBS. After 21 days, continental challenge, and then we follow up for that immunization. And that's what we got. So, the interesting thing about the FCPA is that not only protects against the homologous, which is the Copenhagen, but it also protects 100% against all those different strains that we use. And this is like between different species that are not only silver, but different species. Different species, those are different species. So it protects against them all. And you can see here the heat vaccine usually is done in 2 doses, and even when you do 1 dose, not even the homologous is able to protect against. So highly effective in terms of protection of survival. Unfortunately, what you can see is not very effective in protecting against renal colonization. So it doesn't give you like sterilized immunity. So even with the homologous, it's not, you cannot reach 100%. When you go to it, it gets even worse. So high effective in protecting against mortality, not very effective in protecting colonization. So the issue here is that animals, as I mentioned before, is highly sensitive animal, less than 10 leptospirosis to kill the animal, and when they, for any reason that you have like a disbalance between that pathogenicity, animals become colonized. So it's not a very good model to study colonization. So we decided to test a mouse model, which is a better model to determine colonization. So we saw dissemination. We actually do different doses of SPA, and you see the same pattern. So leptospirosis are, they disseminate in the blood, they disappear, and actually, animals don't get colonized at all. The wild type, they actually, have same pattern, but the animals ended up being colonized, which is different from the FCPA. So yeah, similar pattern that you see in hamsters. So we decided to do the same vaccination. The only difference here is that we use different doses of the vaccine from 10^7 to 10^1, but very similar protocol. And what we saw here, interesting, is that in terms of protection for that, 10^7, 10^5, up to 10^3, all the animals are protected. And this is like a cross protection with like 1 serovar, but cross protection. So, and of course, 10^2 only 50%, and then 10 leptospirosis they cannot protect at all. However, when you do kidney colonization, only 10^7 was able to give you protection, but it did give you protection against colonization. So just showing the promise that if you use a proper model, you might be able to see the results that you're looking for. And that was the case. So although 10^5 to the 3rd give you protection against that, they don't give you protection against colonization. But now you have a vaccine that potentially can protect against that about all the serovar, but also protects you against renal colonization. So, in the when we do like a PCR, you can see that although the enhancer, although you don't have a sterilizing immunity, you do have a reduction in terms of like the burden of the disease in the kidney. The blue here is a PBS, but rather than vaccines, you can see it works very good in terms of the acne, of course, it's homologous, but but it do have a significant reduction. And same thing for the mouse, and 7 of the animals are protected, but then you have a reduction of the dose when you see in the mouse and different doses as well. But the interesting thing about the vaccine is, okay, now we have protection, we have that anyway, so why those animals are being protected? So when we did like agglutinating antibodies, and you know, this is not, it was not a surprise, but agglutinating antibodies only work for the homologous, right? Because that's what I mentioned before. LPS is conserved, antibodies are against LPS, and you know, you can only recognize the same strain. So you know, if you do agglutinated antibodies, you cannot have any titers for any of the strains that you use, although you do have protection. So clearly, agglutinated antibodies is not you know, correlated for protection. What we did see is actually antibodies against proteins that are correlated. So you can see here, if you use the antibodies about those animals that are vaccinated, they recognize pretty much basically the same proteins all over the different strains that they use. It's the same phenotype also in the mouse serum. So really showing that actually antibodies and the proteins are the ones that are correlated with protection. And you know, in using the and we just did like a test with the serum from the mouse, and we basically treated the antigen, either no treated or treated protein escape to eliminate all the proteins. And you can see once you treat it with protein escape, your results in the line is completely zero because you destroy all the proteins. So all those antibodies results that you see here is anti-proteins. So you know, showing really that those antibodies, anti-proteins, are the one that is leading this protection. And you know, and as in lepto, we know also that antibodies are the correlates of you know, of protection. You know, if you do like a massive transfer of those animals that have been vaccinated against the FCPA mutant, you know, all the animals are protected, both in the hamster and in the mouse. So basically, just showing that antibodies, anti-proteins are enough for the protection. So the role of T cells, it's still like a, we don't know exactly what they are, but it's just that antibodies per se, you know, overall response is important. And then later on, actually, we did a bactericidal assay, and you can see that there's high levels of bactericidal activity for the serum of those animals vaccinated against all those different strains that we use, like you know, between like 70 to 100, almost like 100%. The mouse sera 10 to the 7 is actually the same. You can see there's, as I mentioned before, 10 to the 3rd protects against that, and you can see the bactericidal activity of the mouse very high in those 2 doses. We start decaying 10 to the 2nd and basically now 10 to the 1. And also when you dilute the hamster here, almost like only 1% of the sera is still maintained like almost like 100% bactericidal. So there's very high bactericidal activity of the sera. And one important point that you can see here is actually, although it does have a high bactericidal activity, you cannot relate the bactericidal activity with sterilizing immunity, right? Because we know that those animals here, although this high bactericidal activity, they still, colonize the kidney. So there's a disconnection between bactericidal assays and leptospirosis when it comes to kidney protection or sterilizing. I'm trying to work on that. And then what we did, I told you about yesterday about the proteome array. So we use the same system for those few of those animals, and actually we were able to identify which are those proteins that are correlated with that protection. So we were able to identify 155 most expressed proteins based on the proteome array. And this is like an interconnection. We have evaluated separately those animals, the hamsters, then to the 7, the mouse, and to the 7 because it gives protection against colonization. And then all the doses that we use for hamsters, for mouse, sorry. And in a lot of those proteins are automated proteins, and of course a high majority is like a hypothetical protein. That's it. The problem also with lacto. A lot of the protein in lacto, there's a lot of proteins that we don't know exactly the function, and they have no correlation to any other bacteria. FCP1, FCP8 actually was an example. So there's not much to go, and actually we want to dig each one of those. But we thought, okay, so now we have like this potential source for a sub-immune vaccine because we have like very biological relevant candidates, that we didn't have in the past, and a lot of people don't have. And the interesting thing, we got only this intersection here just to give you like a snapshot, but you can see a lot of those, almost 80% of those are highly conserved. The red here is like highly how conserved they are. The green actually there's no those strains here don't have those species here don't have those protein, but it's the vast majority of them are highly conserved among this. So it's like it's another evidence that that's why this vaccine is actually so effective. You can see here that the animals that are not vaccinated have the signal. But the also interesting thing here, if you look at patients with lab, a lot of those proteins are also important in humans, in disease. So just seeing that really showing that this list of candidates that we have already for and biological development. So why a live attenuated vaccine selection would be important. So you have the potential to formulate a whole leptospirosis vaccine with a single strain. Eliciting antibodies against leptospirosis proteins, species, confer cross-protection immune against that against maximal infection. So we have the same antigen which are comparing cross-protection immune and attenuated vaccine model available natural bioimmunity infection in humans. That's what I showed you before with the array. And then you have the potential to formulate our approach for prevention because you can use like different strains for different animals, deleting the FCPA and so far. So the issue is that there's some limitations for human use. This is like this vaccine, this strain has antimicrobial resistant cassette. I mentioned yesterday briefly, but genetic transformation is extremely difficult. Our group is one of the few that in the world that actually can do that. And even us, we still haven't been able to actually create like a completely clean bacteria. We've been working a lot. We've been working with Solaris, trying to make this as a product. But one of the issues has been trying to actually create an XP that doesn't have this model. So that would be a major block for human use. But also we're still very close to what a whole vaccine, whole bacteria, and all the uses that they have in a whole vaccine. So we are still trying to see, okay, can we go further with this? Now we have this list of important biologically relevant candidates, and we just move a step forward in terms of subunit vaccines with that list. As I mentioned before, a lot of the time people do subunit vaccines after they try for those very known candidates that people know might be important for virulence, like leptospirosis, like licks. And although a lot of studies show that those are not good candidates, people keep trying to change using different attributes, using different ways of methods to create a vaccine and trying to, and it's very snowball that people needs to know it. So now we have all those proteins that are interesting, that have potential to be candidates, and we can explore them. So looking at the list and base what we got from the FCPA, using that as a surrogate for acquiring immunity against leptospirosis. So we decided to make a list of what are the candidates that we can actually potentially explore. So a lot of those I mentioned before, 21 of those 154 are autoimmune proteins. So we based on mutants that we have in innovation, based on other studies of proteomics, other studies of transcriptomics, we made a list of 8 candidates that we thought, let's try to see if any of those candidates would be important to look. So you can see here in terms of amino acid identity, there's one here that actually is very bad in terms of amino acid identity, although it is an important protein in terms of pathogenesis and immunizing, you have to show the virulence factor. So we decided to keep it in the list, and there's one more, that's one strain there that they have, but most of them are highly conserved. So we thought, okay, first of all, they're interesting candidates, but when you test them, all of them individually, nothing. All completely die, not even the model of protection. So just going back to the same issue, that it might be very difficult to try to find one protein of leptospirosis because of the redundancy that is going to be able to be that protection. So what we decided to do is okay, so what happens if you mix those proteins, right? So if you put a multi-protein recombinant vaccine. So my goal was to do using a combination of those autoimmune proteins to use cross protection against leptospirosis. So this was completely, there's no rule here for how to do that, but I decided to do, we have 8 proteins, so I decided to do 3 groups of 4. So we have here, this is the list of the 8, we have group 1 here, group 2 here, and then a mix of the group 1 and group 2 would be the group 3 because those are the highly conserved proteins that we have. So we tested as a proof of concept, but just 4 hamsters. we use like those 3 groups. We actually vaccinated animals 3 doses, 40 micrograms of each of the protein, 160 total, in a 2 weeks interval. Adavax was used as an adjuvant, so it's like a swelling oil and adjuvant is better for like a TH1, TH2 response, better than alum. And then we use Ligay as a control, as I mentioned before. Ligay has been shown to be protective of homologous. And also, like, we combine 4 non-autoimmune proteins as a control, and of course, only adjuvant. And then 2 weeks after the 3rd dose, we challenge the animals, congenital without, and we use copiapine as a homologous because that's the sequence of those proteins. And then we also use hardro, which is a very far vorticized species, where the identity go like below 8%. So we thought it was a good heterologous trait to be used. So, when we, after vaccination, when we did after a model challenge, as expected, of course, all the controls died, Ligay survived, no RPE all died as well, but then like all G1, G2, and the 3 groups, they all survived for a model. So, combination of those 4 proteins already give you a boost of immunity, and all those animals survive. But when you do like colonization, we know that Ligay, as I mentioned before, doesn't mean colonization, so animals are colonized, or we colonized. But interesting, all G1, G2, G3, all animals completely stabilized immunity. So very promising for that for those combinations. So then heterologous challenge, is that the same thing? I mean, here actually hardro doesn't have Ligay, so not surprising all the animals Ligay died. But then you see here G2 also died. So it's a group of proteins actually could not give you protection against heterologous. However, both G1 and G2, in G3, actually the animals survived with the heterologous protection. And then the more interesting thing is that when we did qPCR, again, Ligay, of course, all colonized, controls colonized, G2 colonized because they also died, but then G1, although survived, they are colonized. But then we can see here the G3 group is the only one that not only made the animals survive, but also was colonization whatsoever. So, we decided to expand that result. We did more animals. We decided to use different serovars and different species. We have only used one, so only Copenhagen and hardro. So now we use like other examples of Allen's another Cushionaria, and again hardro, 7 animals. And we can see here that there's some interesting results. So the G1 that I mentioned before that was not protected. So let me go back one step. So all animals survived. Okay, so so it proved that both G1 and G2 100% effective in protecting those animals, both homologous, but also all for the other strains. But when we did PCR, the G1 group actually was very good in protecting against all the interlogous strains, but not very good in protecting immunocolonization, because we have seen before how it didn't work, but it also didn't work for Cushionaria, which is another species as well. However, as we see before, the group 3, which is the green here, protects against every single serovar that we use. So this showing that the protection against all of those species as well. And there's promising results in terms of like also sterilizing immunity. So those proteins give a very good immune response, a good boost for all the doses that we use, both for homologous and heterologous. And an interesting thing is that the immune response is dominated by IgG3. There's some groups recently shown that IgG3 might be implicated in renal clearance against leptospirosis. Just to give an example, most of all the studies that have been done in terms of like the vaccines or wholesale vaccines, IgG2 is usually the one that prevails. So that might be one of the reasons why this vaccine is more effective. And then when we did bactericide activity, it's interesting that you can see really the difference in terms of protection. So you can see the G1 group, which is the one that protects against the group, the interlogous, but not against other heterologous. So you can see there's a 40% bactericide activity, a little bit lower on 2, which is the one that doesn't protect at all against infection. And then you can see the O3, has not so high, but 60% overall in terms of bactericides. So can be have a better use for recombinant proteins. You can have a better correlation between bactericide activity and protection, not only against that, but also against immunocolonization. And then we show more recently that those proteins have like an important role in the leptospirosis. So we tested like a group of patients from our active surveillance in Salvador, and you can see here all those 4 proteins have a very good immune response for those patients in terms of IgG in their acute phase, not very good in the convalescent, which makes sense. And then there's a shift, which is very normal in proteins for lacto. You know, the IgGM response for convalescent serum drops, but then you have an increase in the IgG slow in the acute serum, but it then has this higher in the convalescent, which makes sense because there's always a shift in the IgM response. But the interesting thing is that we've been doing a collaboration with USDA that shows that actually the serum from cattle, and this is animals that have been like, you know, the rats, the MAT positive, qPCR positive, and goes from like MAT positive qPCR negative with high titer, low titer MAT, and then animals are completely negative. And it's very interesting that you can very distinguish like the animals that have been either qPCR positive or high titer MAT, and then, you know, lower signals when you have those animals with lower titer, and of course the negative one. So just showing that, you know, those proteins can also have potential for, you know, working diagnostic and differentiate, as I said, as a form between animals that are vaccine animals that are really sick. And you know, just a hint of like you know, as I mentioned before, we do also a lot of work in pathogenesis. We just submitted this paper. This is one of the candidates of the vaccine, just showing how relevant those candidates are. This is like a major pathogen binding protein. It's an outer membrane protein. It has like a spore domain, which usually is believed to bind to active glycan, and it has involved in the division of the cell, but it has a completely different function in lab that will show that actually you know, it's important for adhesion of the cell. So you can see the mutant actually reduce the adhesion of the cells to MDCK, and also it's very important for translocation. So you can see here the mutant is not able to actually translocate MDCK cells. You know, restore when you complement that. And the interesting thing is that it seems to be involved also in degradation of fibronectin and highly binding plasminogen. So just showing there's an important release factor in lacto. So not surprising that it works so well in the vaccine. And then 2 of the other candidates, we also have mutants as well. This one is very interesting. I told you about the whole issue of like route of infection. This mutant, if you do IP, it still kills the animals. But if you do conjunctival, then you survive. So it lost the ability. And if you do like a translocation, you can see that the mutant lost the ability to translocate. So you can only see that, you know, that phenotype when you do conjunctival and not IP. You know, again, another virulence factor, and we have this one is also virulence factor. It's completely attenuated, but we're still working on that to try to understand the mechanism of the attenuation. But just show that at least 3 of those 4 candidates we have mutants, they all attenuation, they all important in virulence. And you know, really show that it's important to have a potential for a universal vaccine. So a lot of those strains that we have in lab, a lot of the species we actually don't have animal models. They don't work very well in animal models, but you know, bactericidal activity is very high also for those strains, including like Santa Lucia has been, you know, found here in Colombia. So you know, but using bactericidal activity is very high still for those strains, especially for the B2 group. So really show that that has a potential for infection. I know we're probably gonna running out of time, but this is like the last slide. I just want to, you know, show that this group is comprised of highly conserved genes and listed 100% cross protection against that, and also 100% sterilized immunity. This is like really the first evidence of 100% protection in, you know, using recombinant protein in leptospirosis. Nobody ever been able to achieve that, and it just shows that with the correct targets, you know, this universal vaccine is really a potentially feasible thing to reach. You know, and it has the potential to a vaccine like this has the potential to against, you know, protect animals or humans against all species of lacto-pathogenic species, eliminate issues of production and safety of current bacteria available for lacto. I mean, those bacteria, they have to be like grown, you know, fermentators, large, you know, batches. There's a lot of discussion regarding like, you know, the media that you use that can change immunogenicity of those vaccines. So you eliminate all that. And then an effective vaccine could be used in livestock, domestic animals, you know, as a blocking vaccine with more health complications, of course, to reduce the risk of major carriers, but also to, as I mentioned before, to be able to differentiate animals continue to do studies on this. It's a project that is still ongoing. So we're going in the direction of trying to not only we have those 4 proteins, so now we want to identify what are the epitopes of those, and then we can do more like a multi-primary epitope vaccine for that. We're also trying to see if we can improve those and also like our interval groups of them. Really trying to optimize the protocol vaccination. And we also be involved with boring eater right now to try to test those vaccines in dogs to see how effective they are. And and try to move a little bit more to the pathological features. And this is just like we literally just doing that. We already did. We have been working with Syrian company in California. They have this like very interesting high throughput method to identify immune epitopes when you use like sera of those animals. We already actually did this. We identified at least 23 very significant epitopes. We literally this week it's a MPHD is from one of my students that we just tested, and all those 6 seem like very highly immunogenic for both animals and humans. So we'd be very excited about that. And as I mentioned before, yesterday we did an extra acquired immunity in humans, and we continue to do that also in alpha-lima. And just to show you how interesting this list of candidates that we have for the attenuated vaccine, we did a case control study in the alpha-lima to see about individual infections, to see if those antibodies against proteins might be related to protection against infection. And we were able to identify 37 proteins that both IgM and IgG. And at least 18 of those are correlated to the attenuated vaccine as well. And we actually tested 10 mutants among those 37, and at least 60% of those were attenuated. It's showing like how this list is really important for us to determine and be used as both of the diagnostic vaccine. So again, a lot of people involved, but the whole goal is try to protect this is called Lima, the kids in our community, and to try to make a vaccine that can actually protect this population from disease and from that. So, thank you. Question or
Yeah, I wanted to ask you a little bit more about diagnostics. So you showed the efficacy of 2 types of tests, the days after onset disease. At what point after onset do people typically get sick enough to seek diagnostics?
Well, so I think that goes back to what I discussed again. So it varies a lot. So you have people who go to the hospital with like 3 days of disease and are ready with like very acute symptoms, and it actually immediately goes to like UCI or ICU. And then you have patients who come like maybe 7 days of disease and continue to have only fever and go for basically no treatment and go to resolve itself. So that's the part that we don't know, because it might be involved with those of the disease, or might be those of infection, or might be involved with with how some genetics of the patients. I mean, the average that we have for individuals that go to the hospital when we have our active surveillance is basically like 5 to 7 days. That's the average that you have. But you have, have patients that goes even later than that, with a fever as well. But consistently, we saw in all those exercises, we saw both for the dual platform, but also for PCR, is that earlier, it's better, especially for the PCR because the blood. But for the treatment, it's important that you have a test that is good in the early phase of the disease, so that you can treat those patients. Because a patient that gets there and goes to ICU, again, the chances of those people survive just drops to like 20%.
How often, if diagnostics were widely available, how often do healthcare providers even think about leptospirosis until the ICU phase of the disease occurs?
That's a very good question. In most of the cases, they don't. Yeah.
They think it's dengue or something.
Exactly. Yeah. So that's what I showed you yesterday. A lot of the cases in solid, for example, of leptospirosis, recently diagnosed as dengue mostly. I'm pretty sure that if you go to Africa, there might be some plantation and malaria. So, yeah, lab is not even like in the top of the line. Veterinarians are better for lab. It's more. Well, maybe not. Well, but in general, I'm just thinking that's a very important disease that we talk. It has economic impact and things. So as veterinarians get out of school knowing about lactose, and if you talk to AD, most of the time they just barely know exactly what. So education for especially for those areas where lactose is endemic. I think Thailand did a good job in that. They have like an increase after 2004. They have like a lot of outbreaks there, and they made like a very intensive training and health education for the providers. And so then you can see a spike in terms of like diagnostic, in spite of interest in testing, because they did a very good job trying to talk to people to think about leptospirosis before they at the same time they think about other diseases, but thinking about leptospirosis.
I'm sorry, but
No,
the way that entonces las otras preguntas fuera con él. Gracias por haber venido.
Muchas gracias. Sí,